morning, everyone. Welcome to our final session for exercise 7. So today is our last session for for the whole exercise session, and we'll be working through a gene expression data. So here are already some tips that are on the screen for how to import the data. But first, let's take a look at the questions first. So it's a quite a long one. There's some introduction on the data set. First, we have this gene expression data from 5 different tumor types. So, and the tumor types are specified in the CSV file. So the data represents how strong each gene was in each sample. And the idea is that different tumors have some overall differences in gene expression, making them distinguishable from each other. The dataset contains 795 samples, each with 2,257 features. And the data is already pre-processed by removing features with small amount of variations, as well as some invalid rows. So your task for today is to build a system that can say from which tumor the given gene expression data came from, which means you're building a classifier to classify between tumor types. And can you reliably separate classes from each other? And the second task is to study which of the gene expressions are important for the classification, which genes separate the tumor, which means which genes separate tumors the most, and provide your arguments for your conclusions. Can you find simple rules like if X expression is higher than Z, then sample is from tumor Y. And you can identify genes with a column index in the given data set. So for our task today, you can use any existing implementations of algorithms. For example, you can use scikit-learn, PyTorch, or you can use any libraries in Python. And you're also free to use any techniques presented during lecture or found elsewhere. However, you should report all the steps and techniques that are used in your work. Here are some tips and hints. Let's go through them first. First column in the expression identifies samples from each other. Same with the CSV file. Note that they're not aligned. Row X in the first file may not be the same gene as row X in the second file. And let's take a look at the data sets. First, in the gene expression data, we can see we have different samples. This is the first row with sample 0, and it has many features. They are space separated, and from the questions, we can see each has around 2,000 features. So the first column is the sample name, and all the rest columns are the features, and each one has around 2,000 features. And from our first hint, we can see that these are not aligned with the label CSV file. because if we move it to the end, we have 795 rows, but the sample number goes up to 800. So, and it's also pointed out that this data set has already been processed before, so some invalid rows are removed. But for the CSV file, we have our sample ID and the class the sample ID belongs to. But for our CSV file, we have the correct number. We have all the classes for all the sample ID up until 800. Because if you remove 801, because if you remove the header, there should be 801 sample IDs in total. So for gene expression data, we have some rows removed already, and that you need to take that into consideration later when we're importing data, because we we need to match them to the sample ID and class in the second file. And to get a general sense of classability of data, you can start clustering the data set with different algorithms. For example, you can use k-means or any other algorithm you think is suitable. And to visualize the high varidimensional data, you can try dimensionality reduction. The core idea is to turn high dimensional data to low dimensional, usually two-dimensional for plotting, while also try to preserve the structure of the data in lower dimensions. You can also turn two-class classifier perceptron into multi-class classifier by training one classifier for each class, or training one classifier per class pair, taking the majority vote of the classifier. And the difference between these are called one versus rest and one versus one. Another hint is that gene expression may be highly correlated with each other. You can study relation between gene expression and tumor types by visualizing and studying the distribution of features or by studying the weights parameters of learned models. You can also try to brute force the problem. By definition, if the feature is not important for classification, it shouldn't affect the classification result. And our data is from, here are the source for the gene expression data that we got today. It's linked in the PDF file. And the data set that we have here, like it's suggested, it has a very, very, very high dimension. It has around 2000 dimension for 2000 dimension for each sample. So, however, for the class, here we can see clearly we don't have that many classes. We don't have that many classes here. We can see we have overall. We have overall 1, 2, 1, 2, 3, 4, 5. We have 5 different classes. We have 5 different classes in total. Okay, to keep that in mind, then we can start the implementation. So I have already imported all the libraries we're gonna use. It's mainly NumPy and Matplotlib and different smaller components from scikit-learn. So we'll be using PCA, and it's a function called principal component analysis, and we'll give some introduction to it later. And we'll also be using perceptron, We have implemented this ourselves before, the perceptron classifier, but today we're gonna just import it from an existing library, trend test split. We also have given some introduction to this last time when we were training the neural networks. Also a standard scalar. This one is used for standardization, or you can also call it normalization. We have implemented the normalization algorithms ourselves as well before. And today we're going to use this standard scalar from the library. First, we need to import the data set. So we're going to use first import the expression data that's in the tips from the slides. We're going to import it like it was suggested on the slides. So we imported this expression data, but we chose all rows, but we only start from the second columns because we want to remove the header and just keep the gene expressions. And then we initialize an empty list. So we initialize an empty list. Then we will hold this list will be used for holding the tumor types for each sample in the order of the expression data. But then we need a dictionary to store this mapping from sample ID to tumor types. And to do that, we need to initialize a dictionary as well. And we'll call it map sample to type. And this dictionary will be used for storing the mapping. Then we need to import the CSV file. First, for the CSV file, we file read line. This basically means we are skipping the header line of the CSV file. So we don't need this. We are skipping this line, the headers. And then we loop through each subsequent line in the CSV file. Then we use line strip. We use line strip to remove any trailing white spaces. For example, if there's a new line characters from the line, then we remove them. And it splits the line by comma. And the limit is to one split. So so the lines here, they get to split into two variables. Then we add an entry to the dictionary. The sample ID is the key, and the type string is the value. And then we open the gene expression data again. And because the gene expression data is space separated, it's separated by space. So here we're splitting them by space instead of comma. And then the type list will contain the tumor type strings, and the order of these strings will perfectly match the order of the samples in the data NumPy array that was loaded earlier. That's why we loaded them twice here, because the first time we are loading the data, and the second time we create this dictionary to map the order of the data because it's not perfectly matched to the CSV file. And there is some redundancy here. Can you make it more efficient if you only load it once? Yes, there are more efficient ways, but this is the most, this is a very simple, straightforward way to do this. So after we have fully loaded the data, then we need to convert the classes, the strings here, because they are stored as strings in the dictionary, and we should convert them into numerical labels because most machine learning algorithms require numerical input for labels. So this is a very standard pre-processing steps. So we loop through all the types. And if type here is giving some suggestions, but it's not fully correct, so we'll do it ourselves. So the first one is PRAD. And we'll give it a label 0. And for BRC, is the second one BRCA? No, it's the LUAD. Then we give it a label of 1. Then it's BRCA, that's right. Then we give it 2. Is this one LCSC? No, it's KIRC. That's our fourth label. And we give it a numerical value of 3. And the last one is COAD. That's correct. Yeah, COAD. From 0 to 4, and we have 5 different types of tumor types. And we have converted them into numerical values. And now we convert the types list to a NumPy array. And we can set them explicitly to set the D type to int because we're using integers here. Then we can initialize our PCA to reduce to two components. PCA we have discussed it earlier. We're just calling the library. So we set the components to two. Here is some, they give you some general tips about PCA. And what PCA is called principal component analysis. So it's an unsupervised dimensionality reduction algorithm. And what is dimensionality reduction? So imagine that your data has many features or many columns. Well, in our case, we don't have to imagine because we have a very high dimensionality data set. We have 2,000 features. And in this dimensional space, in this 2,000 dimensional space, it's impossible for us humans to visualize this directly. For example, if it's one-dimensional data, you can draw it on a line. If it's two-dimensional, maybe you can draw it on a plane, and three-dimensional, maybe a hyperplane. But higher than that, if it's fourth dimensional or higher dimensional, for example, this one, it has 2,000 dimensions. And for us humans, we are unable to visualize this directly. And it's also impossible for us to plot this directly. So what PCA does is reducing this high number of dimensions, or you can also call them features, to a much smaller number. For example, you can reduce it to 2 or 3 for visualization. But during this process, it's still preserving as much as important information or variance in the data as possible. And a very important part is that this is an unsupervised algorithm, which means that it works without looking at the class labels. It only looks at the gene expression value themselves and try to find this inherent structure or patterns within the data set. And so maybe we can talk about how it works. So we can imagine all of our data points are in this. We cannot visualize it directly in our head, but we can just imagine all these data points as a cloud that has 2,000 dimensional space. And the PCA will try to find a set of axes. And those axes are called principal components. And they will try to find a new set of axes to represent this data. And for us here, we have set the PCA, the principal component, to 2. So basically, it means it will find they will find 2 principal components. And for our first principal component, the PCA will try to find the direction in the data that has the largest variance. And it will be like this line you could draw through the data cloud, this 2,000 dimensional data cloud. And it will be this line. And all the points are spread out on this line. And then you will project this data onto this single line to capture the most significant data of the variance. And then for our second component, the PCA will find another direction that is perpendicular to the first component, which means that it will go at the first component at a 90-degree angle. And this line will have the second largest variance among all possible other perpendicular directions. And if you set it to higher to 3, then the algorithm will try to find subset principal component. But for our implementations here, we'll use 2 principal components. And so after we have selected 2 principal components, then we projected our high dimensional data onto this lower dimensional subspace. Then we need to fit the data. So to fit the data basically means the PCA will learn the principal component from this data. Then we can start our 2D PCA visualization. First, we define different colors for different classes. Blue, maybe yellow, green, and black. Okay, just to keep it consistent. So then we can start to plot our figure. And we will enumerate through all the tumor types. So the first one is PRAD. And the second one is LUAD, BRCA, KIRC, and COAD. And first, we need to select the data points belonging to the current tumor types. So the data 2D here, it will use the Boolean array. Sorry, I think the connection dropped for a second. The Boolean, the 2D data here, you use the Boolean array to select rows from data 2D, and only rows in the data 2D where its corresponding value is true will be selected. Then we can start our scatter plot. Is it correct? Yeah, it looks correct. First, we're selecting all the rows from the data, starting and the first column, all the rows and first column. These are the x coordinates. For all the sample of the current tumor types. And these are the y coordinates for the current tumor types. Then we can add a legend. Also show the plot. Okay, we can see there's some overlaps here from tumor type COAD and LUAD, the blue and the black ones. So we can add an alpha value here to make it a bit transparent so we can see the overlapping. And also maybe we make the figure size a bit bigger. Okay, now it's more clear. And by now we can already answer some of the questions. We can actually already answer some of the questions. Can you reliably separate classes from each other? And we can see there are some classes here. The green ones, the yellow ones, the red ones, they are very, very easily separable. They are very far from each other. The blue one and black one, they are kind of close and they may seem to have some overlaps here, but I think it's not so overcrowded together. But to continue the of building our classifier, of building our system that can say which tumor type is from which. And let's continue. This is a visualization of using the PCA. And we can have a overall idea of what the data set looks like. So next, just our usual procedures for neural networks. We are splitting our data sets. We have data train, data task, type train, type test, using the library from train test split. And we're using this as data types. Test size, we set it to 20%, which means we're splitting our data sets into two parts, and 20% of the data will be allocated to the testing set, and the remaining 80% will be used for training the model. After we have split the data, then we need to standardize it using the standard scaler. And we fit the data using the standard scaler. Then we need to, after the fit method calculates the mean and the standard deviation for each feature, which we have implemented ourselves before. So we know the algorithm, the formula behind it. And here, the first one here, when we're fitting the data, we're using only the training data. And that's very important to prevent data leakage from the testing set into the training process. And when we are transforming the data, which means we're applying the calculated scaling to the training data itself. And then we also need to apply it to the same scaling transformation to the testing set. And here we are subtracting the mean and divide the standard deviation that was learned from data train. And then we need to create a perceptron instance. We can set the parameters here. The max iteration maybe is 50, and the tolerance we can give it a 0.001. And a random state here is used for reproducibility. So we can reproduce the result. So for scikit-learn, perceptron itself is a simple linear classifier. But for multi-class problems like this, we have five tumor types. So it's a multi-class problem. And scikit-learn will have by default you will use one versus rest or one versus all strategy. So it means it will train one binary perceptron for each class against all other classes. And the tolerance settings here, we have also explained it before. It will stop if the improvement in the loss function is less than this tolerance after the iteration. So it's an early stopping criteria. Then we can fit the model. We are training the model using the scaled training features, the standardized training features, and their corresponding true labels. The model here, after the model is trained, it will learn the weights that define the decision boundaries between classes. And after now the perceptual models have been trained, we can start to make predictions. So as you can see, after we made the predictions, we transform the data test from its original feature space in a new lower dimensional feature space using the PCA. And after that, we can start plotting the test data. I think we forgot to define it earlier. We can define it again. We can define it again, the tumor types. PRAD, LUAD, BRCA, KRC, COAD. So we can use it later. We should have defined it earlier, but we can use it. This is correct. We will loop through all the tumor types that we have here, the 5 tumor types. Then for each time, we will select the 2D test data points where the predicted class for the current number, which is I. This is similar to before. So, but here we need to make some comparisons. Because we will only attempt to plot if there are actually any samples predicted for this class.
Mikä siellä punaisuus on tiellä? Ei näy mitään. Ei näy. Ei näy mitään kuin punaisuus. Punaisuus on tiellä.
Okay, so we're using basically the same implementations as before. We have our x-axis, x coordinates, which is the first principal component, and the y-axis, which is the second principal component. The colors are based on the colors we have defined before for each tumor types. Then we will have this label that's for legend and the alpha value set to 70%. Okay, after this loop, then we can give it a title. Predicted class for test data. We add a legend and show the result. So this is the predicted. Does that? Okay, I will mute the microphone if you do not have any questions. So if you have, you can also you can send them in the chat. So for the prediction, we can output the accuracy. We want to know the accuracy. And we can see our accuracy is 99.37%. Okay, that's a very, very good accuracy result. It's uh, it's almost perfect. And if we change our random states, sometimes you might get even higher accuracy. Here we have a 42, right? Uh, let's not set that. And now we have 100% accuracy because the random state also affects the training, affects the reproducibility a little bit. So such a high score, you always in machine learning, you always need to be a little bit skeptical about such high score, whether it's 100% or it's 99.3%. Because sometimes you need to consider whether your data is overfitting or not. But for our data, we know that the data set it could be linear separable. So that's why it could produce this perfect result because the perceptron is a linear classifier. And if the data points belong into the different classes of tumors here can be perfectly separated by straight lines. But of course, or like hyperplanes in higher dimensions. So the perceptron is designed to find such a separation. So our plot uh is in PCA, is in principal component plot. They are not absolutely perfect. We can still see there are some overlaps here in the 2D plot. So it is very possible that in the full dimensional space, which is the 2225 dimensional space, the classes in our set happen to be perfectly linear separable by this hyperplane that was learned by the perceptron. However, when we reduce it to two-dimensional plot like this, we see some overlaps here. Also, there could be that's like the optimal scenario. There could be other situations that could contribute to this very, very high accuracy score. Maybe there's some data leakage or some errors that that was not implemented correctly. So sometimes you need to do cross verification into cross validation. Sorry, you need to do cross validation. Because a single test transcript can sometimes be lucky, and you can try different test sites. For example, you can try 30% or 40%. And we can see when we tried the 30%, we got a still high score, 99.6 almost. So after even we have tried many different parameters, it still gives you this high score. So we can, by now, I think we can say to answer the questions, we can say that we have built a system that can reliably tell which tumor came from given the gene expression. And for our second task, which of these genes are more important for classification. Provide arguments and reasoning for your conclusions. And we're gonna implement the second task in 4 minutes. We can take a break and let's continue at four in a few minutes. We can take a break. And in the meantime, if you have any questions, you can prepare them and ask me later. Let's take a few minutes break. Okay, we're back. Please let me know if you have any questions. So for our second task, study which gene expression are more important. If the perceptron that we have built here achieves 100% accuracy, its learned weight can give us some insights into which genes are more important. So what we are basically asking is for each tumor type, can I find this specific gene whose expression level is good enough to predict whether a sample belongs to this tumor type or not? So let's first get the weights from the original multi-class model that we have built. And in the perceptron coefficient here stores the weight. A larger positive weight for a gene in the row means high expression of that gene, which will push the model towards classifying a sample as that specific tumor type. We can take a look. So we have all these different values. These are the weights that the models have learned. A large positive value, like for example 8 or 9, uh, it pushes the model towards a sample towards a specific tumor type, and a negative number, a smaller number, do the opposite. So then we need to find a promising single gene for each tumor type. That's why we need to find the number with the highest value. So max weight here we're using the argmax to find along the axis of 1, which means to find each row. It finds the column index where the weight is the largest. And here we can see we have 1, 2, 3, 4, 5. We have 5 largest genes for each tumor, for corresponding to each tumor type. Then for each of the 5 tumor types, let's first create a for loop. Because we have 5 tumor types, so we iterate 5 times. We first get the promising gene for the current tumor type, which is here is the T. Then we create binary labels. So it means the current tumor type and not the current tumor type. And to do that, we need to do it for the training set and the testing set. We are populating this array of all zeros in the original shape. When you call it binary. And we set the label to 1 when the we set the value to 1 when the label was t. And then we do the same for testing sets. Okay, so now we have converted our multi-class problem into a series of two-class problems. Because now we are comparing one single tumor, this gene for just one single tumor against all other tumors. So we're not comparing. Now it's a two-class problem. And then we need to get data for only selected single gene. And we do the same for testing. So here we're extracting the expression value of only the chosen gene, which is the index gene for rule. Here, the chosen gene. For both training and testing samples. And then we reshape it into a 2D array. Even though we only have one features, but we still need to reshape it into a 2D array because that's what scikit-learn expects. And then we can train a new simple perceptron on this single gene. And we use the same parameters as before, max 50 iterations and tolerance of 0.001. Then we fit the model. Now the perception object will perception object will is created inside this loop, which means each tumor type will get its own dedicated simple model because we're still in the loop. So we're doing this 5 times and each tumor will get its own model. For our single gene test data, these are the expression values of only one single gene for training. And these are the labels. After that, we can after we have trained the model, or fit the model, then we can make predictions with single gene model. Then we can calculate and print accuracy of the single gene model. Then we print out the accuracy results. Tumor type using only this specific gene. And now we get this accuracy result we get this accuracy result using only a single gene. You can tell how accurate whether it belongs to a specific tumor type. But if we want a simple rule, like if gene X expression is higher than something, then then we can go a step even further, because for a single feature perceptron, the decision boundary, we have also discussed this before when we were implementing the perceptron. The decision boundary is the coefficient that's multiplied by X and plus the intercept, and it equals 0. So if we want to calculate such a formula, we can also implement it. But first, we need to check if it's we need to avoid division by 0. So we need to check if the coefficient is is equal to 0 or not. The threshold is the X in the formula. And the formula, we have discussed it before. After simplifying the formula, then it's minus the intercept that's divided by coefficient. So if the coefficient is positive, then higher gene expression leads to predicting tumor T. Then we print gene expression is a positive. Maybe we'll use a different wording. If gene expression that's larger than threshold, then it's likely then the tumor type is likely okay. Now we have all the results. Now we have all the result, and we have this simple rule. If this gene expression is larger than 1.08, then the tumor type is likely 0, which is the first we have defined it before, which is PRAD. Then it's very likely to be 0, because also the accuracy for this classification is 99.7037, which means we can predict this tumor just based on this single gene. The coefficient. So the overall accuracy, but we also see some discrepancy of the accuracy here, because the overall accuracy earlier was 100%, right? Yeah, it was 100%. And now we see uh some variance in accuracy. For example, here we can we actually don't have we have 100% accuracy, but for tumor type 3 and tumor type 1, the accuracy is much lower. However, even though we achieve very high accuracy overall, that's because for the multi-class perceptron, we have so many more features. But for this two-class perceptron, we are using only one single gene to make the prediction. So, which means some of these genes they were chosen because they had the highest positive coefficient in their respective classifiers. And which means within the context of all other genes, an increase in their gene expression strongly pushed the model towards classifying this sample as that particular tumor type. But for maybe tumor type 3, it's not that important. That's why the accuracy is lower. Okay, now I think we have successfully implemented and answered the tasks. We have built the system, and we reliably separate different classes from each other, and we can identify genes, the most important genes. And we found this simple rule for the gene expressions. And that's it for today's session. Please let me know if you have any questions regarding the courses and the exercises. The exam is on Friday from 12 to 4, if I remember correctly. And the deadlines for submitting all of the exercises is the night before. Because we also need to grade assignments, and we would like to publish your overall grade. We need to take the grade from your assignments into consideration for your overall grade. And so please let me know if you have any more questions. Okay, if you don't have any questions, then that's it for this semester. Lectures and exercise sessions, they have all been concluded. Good luck on your exams, and please don't hesitate to reach out if you have any more questions. Thank you. Thank you for participating in this semester. And also, if you have any feedbacks, there will be a questionnaire. Please fill them out. And if you have any feedbacks, you can also send them directly to either me or Professor Gao. Thank you for participating and for coming to the exercise session. And that's it. Thank you.